Very good evening to one and all. So, yes, so far we have dealt with what beta? Prokaryotic cell is done, cell theory is done, cell discoveries are done. From eukaryotic cell we have completely discussed about cell wall, cell membrane and then endomembrane system, fine. So, today we are left over with these concepts from cell the unit of life, and the concepts include what? Ribosomes, ribosomes, centrioles and centrosome, they come under non-membrane bound organelles, okay, they come under non-membrane bound organelles. Fine, and then cilia and flagella, which are the motile structures, and then cytoskeleton, where we will be discussing about microtubules, microfilaments and intermediary filaments, and then we will be exercising about Raghav, sit here or there or any other bench, and then double membrane bound organelles, which includes mitochondria, chloroplast and nucleus. So, these are the things we have left over. In nucleus, after looking into the structure of nucleus, we have to deal with structure of chromosomes and then shapes of the chromosomes, clear, right? These are the concepts we have to look into it. So, let us get started with first ribosomes, which are non-membrane bound organelles. So, you know ribosomes are present both in prokaryotic cells and eukaryotic cells. First we will once discuss or once remember about the location of the ribosomes both in a prokaryotic cell and eukaryotic cell. If in a prokaryotic cell, if you have to take beta, where ribosomes are present? Ribosomes are present inside the cytoplasm and adjoining to the cell membrane, okay. So, those ribosomes are of what type? 70S type of ribosomes, and larger subunit is 50S, smaller subunit is 30S, fine. Those ribosomes which are present inside the cytoplasm are purely involved in protein synthesis, and those ribosomes which are adjoined to the cell membrane are also helpful in protein translocation, clear, right? Next, where are the ribosomes present in a eukaryotic cell? That is the question. Where are the ribosomes present in a eukaryotic cell? First, over the extensions of endoplasmic reticulum, ribosomes are present, yes or no? Yeah, and inside cytoplasm you can see ribosomes, fine. And if you can take the mitochondria, inside mitochondria ribosomes are present, and if you can take the chloroplast cell organelle, chloroplast, inside chloroplast also you can see the ribosomes. So, here in a eukaryotic cell you can see both the type of ribosomes, both 80S type and 70S type, both 80S and 70S type of ribosomes are observed. 80S type of ribosome you can see in the cytoplasm and adjoining to the endoplasmic reticulum, where you can see beta inside the cytoplasm and which is adjoining to the endoplasmic reticulum. 70S ribosomes is observed both in the chloroplast and mitochondria, fine. 80S ribosome, larger subunit is 60S, smaller subunit is 40S. 70S ribosomes, larger subunit is 50S, smaller subunit is 30S. Apart from this, you have to know what is the composition of ribosome, okay. Ribosomes are called as ribonucleoproteins. What are they called as beta? Ribonucleoproteins. Wherein it has both RNA and amino acids. Both RNA and amino acids are present. These are called as what? Ribonucleoproteins. Fine, right? So, how many different types of proteins are present? If that question comes, what should be our answer is, we have around 80 different types of proteins. Different types of proteins present inside the ribosome. Fine, clear? Yeah, And here one more major point you have to understand, that the larger subunit over 80S ribosome is 60S, but smaller subunit is 40S. How come both together just 80S ribosome you got? What is S in that? What is S over here? Swedberg unit. What does this unit refer about? What does this unit refer about? Swedberg unit. It refers about what? Centrifugation value. Very good, Raghav. What is centrifugation value? It gives about it gives information on what?
On what we have to keep the settings on the centrifugation machine.
So yeah, you are almost there, but let me explain you clearly what he is saying. Like, so to know the value of any organelle that you want to know from the cell, what you do is sedimentation. Sedimentation process is done through centrifugation machine. Okay, so what happens is after centrifugation, the sedimentation value for the larger subunit you got at 60S, smaller subunit you got at 40S. But the sedimentation coefficient after the centrifugation, the sedimentation coefficient which you got for the entire organelle is 80S. Why is that happening is because of the association between larger subunit and smaller subunit. The association is decided by the concentration of magnesium ions. Fine. The association between larger subunit and smaller subunit of the ribosome is decided by magnesium ions. Less the magnesium ion concentration, there is a dissociation between larger subunit and smaller subunit. Fine. More the magnesium ion concentration, more is the association, less the magnesium ion concentration, dissociation between larger subunit and smaller subunit can be observed. Understood, right, ma? So everything is clear about ribosomes, no? Now, here you have to also look into a condition called as polysome. Polysome, which is observed only in the prokaryotic cells. Fine. So, during the time of translation, what happens, ma? To the mRNA chain over here, to the mRNA, ribosome will get attached like this. And over the ribosome, what happens? The tRNA will come and sit at the P site of the ribosome, will copy the codon, and the anticodon will be entered by the tRNA. Yes or no? This is the process of translation. So, at this time in a eukaryotic cell, what happens is, to a single mRNA, one ribosome will be attached. Once the tRNA comes, copies, it leaves, right, then one more ribosome will come and attach. As a result, you will get a monocistronic unit of a protein chain. Okay, monocistronic unit means single peptide chain you will get. But that is not the case in the in prokaryotic cell. What happens is, to a single mRNA chain, many ribosomes will come and attach. Multiple ribosomes will come and attach, which gives you polycistronic expression of proteins. Okay, what expression, ma? Polycistronic expression. Polycistronic protein expression. Understood what is polysome? What is polysome? Single mRNA adjoined with many ribosomal units is called as polysome. This is observed in prokaryotic cells, which gives multiple protein expression, that is polycistronic protein expression. Clear? And in eukaryotic cells, what is observed better? Monosome condition. Eukaryotic cells, monosome condition is observed. Yeah, monocistronic expression. Clear, right? Understood? Okay. Next, centrosome and centrioles. So, ribosomes, which are non-membrane bound organelles, those are observed in prokaryotic cell, eukaryotic cell. But then centrioles or centrosomes, these are observed only in the animal cell bit. Centriole is observed only in the animal cell. So, if you have to consider this as an animal cell, Inside animal cells cytoplasm, what happens is centrosome is present. Okay, centrosome is present, which has two centrioles which are perpendicularly arranged like this. Fine. Centrioles are cylindrical structures which are perpendicularly arranged inside the centrosome. As each centrosome has two centrioles, you can also call it as diplosome. Diplosome. Fine. So, each centrosome is having two centrioles, right? And the centrosome does not have any membrane, it is just having pericentriolar space. Pericentriolar space. And what for this centrioles are helpful? Centrioles are helpful in spindle apparatus formation. Centrioles are helpful in spindle fibers or spindle apparatus formation. Fine. And they are helpful for what? Cell division. Clear? So, now let me ask you a question, that in plant cells there are no centrioles, there is no centrosome, but then cell division is happening. How the function is compensated? How there is no formation of spindle apparatus? You can see spindle apparatus during plant cell division as well. Then how the function is compensated in plant cell? In plant cell you will be having microtubular organizing complex beta. In plant cells, what is present? Microtubular. Yes, beta. Microtubular organizing complex. MTOC. Fine. This basic description is clear to you, right? Now, when you take the transverse section of centriole and see, you can observe the microtubular arrangement in the centrioles. So, if you see the microtubular arrangement beta, at the center there is no microtubules at the center. At the center there is just a proteinaceous hub. Okay? At the center what is there? Proteinaceous hub. To this proteinaceous hub, you have 9 radial spokes attached to it. How many radial spokes? 9 radial spokes. To each radial spoke, peripheral 3 microtubules are present. You can say A, B, C. A, B, C. So, it has what? CA connections. By what protein? Nexin protein. Fine. So, here if you see, in centriole, at the center what is present? Proteinaceous hub. From this proteinaceous hub, 9 radial spokes are radiating out. Listen first. 9 radial spokes are radiating out. To each radial spoke, 3 peripheral microtubules are present. You can name them as A, B, C, and they have CA connections. These protein connections are called as nexin. Fine? Understood, right? So, here 9 radial spokes are there, and at the center there are no microtubules. So, this arrangement of microtubules is called as 9 plus 0 microtubular arrangement. And if you can see in a one centriole, how many microtubules are present? In a single centriole, how many microtubules are present? Total 9 radial spokes na beta. To each radial spoke, how many microtubules are present? 3. So, total 27 microtubules in each centriole are present. Clear, right? This is the complete description of centriole. Same question if I put, in a single centrosome, how many microtubules are present? Simple. Centrosome has 2 centrioles. That's it. Very good, Rahul. It is 54 microtubules. Clear, right, ma? All of you? Yeah. Just I will give you 1 minute, go through this. Any doubts anywhere you can ask me. Done? Yeah. Now, so centrioles and centrosome is clear to you. Now we have to see cilia and flagella. First we will go through flagella. Flagella and cilia. So in flagella we can see the motile structure flagella both in the prokaryotic cell and eukaryotic protozoans. Okay? Be it in the flagellated protozoans or euglena, right. So, in prokaryotic flagella, what type of microtubular arrangement is seen? 9+0 arrangement, and the protein present is flagellin. Fine. Whereas in eukaryotic flagella and cilia, eukaryotic flagella and cilia, the microtubular arrangement is 9+2. I will explain what is 9+2 arrangement now. And then it has, flagella is having what protein beta here? It has dynein protein, not flagellin. Okay? It has dynein. Sorry, it has tubulin protein. It has tubulin protein. It is the filament which is coming out from the basal body, right. So, if you consider this as cell membrane, flagella will be originating like this. It has basal body filament hook. This filament is made up of protein, and here in eukaryotic flagella it is tubulin protein, here it is flagellin protein. Understood? Yeah. Now you see this 9+2 arrangement. Any other doubt, ma? Here you see at the center you can observe two central microtubules at the center. These two central microtubules are covered or capsulated by a central sheath. Okay? Two central microtubules. So, from this central sheath 9 radial spokes are radiating out. Yes or no? How many radial spokes? 9 radial spokes. Each radial spoke is having 2 peripheral microtubules. You can say it as AB. So, total how many AB connections here? Okay? And this is by the protein dynein. Fine. So, here in this 9+2 arrangement, if you have to see 9+2 arrangement, how many total microtubules? At the center you have 2 microtubules plus 9 radial spokes. Each radial spoke is having 2 microtubules. So, total how much, ma? 20 microtubules on total. You can see in eukaryotic cilia and flagella. Understood? Any doubt anywhere? No, right? Take down quickly, 1 minute time I will give. If you are having any doubt, you can ask me. Here, connections between 2 peripheral microtubules. Yes. Done? Next. So, here can you see, ma? Dynein arms, radial spokes. So, this is the TS, where you can see radial spokes, dynein arms, central microtubules and peripheral doublets. Okay? So, here if you observe in eukaryotic cilia and flagella also, first we will see about flagella. Let us suppose this is cell membrane. Cell membrane is coated by what? Cell wall. So, this Flagella and cilia are originating from cell membrane. They are originating from cell membrane and they are extensions of the cell wall. Fine. So, here at originating from cell membrane, they will be having rings, LPSM rings we will be saying. They are having what? Basal body. It is present in the basal body. And this is hook filament. Basal body is attached to the cell membrane by centrioles. Okay, with the help of centrioles only, basal body is attached to the cell membrane and cell wall. So, the microtubular arrangement over here will be 9 plus 0 only beta, whereas hook and filament microtubular arrangement will be 9 plus 2. Understood? In general, if they are asking you what will be the microtubular arrangement in eukaryotic, cilia and flagella, your answer should be 9 plus 2. Okay. If in prokaryotic flagella, your answer should be 9 plus 0. In specific, if they are asking you basal body, hook and filament, then you have to go basal body 9 plus 0, hook and filament 9 plus 2. Okay. Now,
Prokaryotic is 9 plus 0.
yes, yes. Now, cytoskeleton. Cytoskeleton is nothing but the name itself is saying now beta, cyto means what? Cell. Skeleton means shape. Okay. So, cytoskeleton includes a network of proteinaceous filaments which will build the proper shape of the cells, and it will promote the movement both in intracellular movements, extracellular movements are promoted by those proteinaceous filaments. They include what? Microtubules, microfilaments, take down parallelly, microtubules, microfilaments and intermediary filaments. Fine. Microtubules are long rod shaped structures which are having what protein is tubulin protein, whereas microfilaments will be having actin proteins, intermediary filaments will be having lamin proteins. Okay, intermediary filaments will be having lamin proteins. So, this is the protein comparison. Now, we will look into the clear differentiation, comparative analysis between microtubules, microfilaments and intermediary filaments. Look at here, ma. What are microtubules? How can you describe them? Unbranched hollow tubules of protein tubulin. Okay. So, they are made up of the protein tubulin. Now, what are the major functions included by microtubules, microfilaments and intermediary filaments? Questions are put either from the protein composition or from the functions. Okay. So, look at it carefully. Functions include, they constitute of, okay, they include constituents of spindle fibers, chromosome fibers, basal body, flagella and cilia. They function as cytoskeleton, that is, it is giving proper shape to the cell and it is focusing on intracellular transport. Any intracellular targets are there, right? For transport of that, microtubules are helpful, and this controls the shape of the wall-less cells and nuclei. Okay. This helps in the movement of nuclei during cell division and helps in formation of cell plate in plant cells during cytokinesis of plant cells, and it also aids in cell movement. And then here if you see microfilaments. Microfilaments are narrow cylindrical rods or protein filaments which are made up of actin. Okay. And what are the functions included over here? Cytoplasmic streaming. Microfilaments will give proper support to the cells and formation of myofibrils. What are myofibrils beta? Muscle fibers. These are myofibrils, and formation of microvilli in small intestine and formation and retraction of pseudopodia in some amoeboid protozoans like entamoeba, and then endocytosis exocytosis movement in paramecium, formation of spindle apparatus during cell division and movement of cellular components. These are the functions of microfilaments. Lastly, intermediary filaments. Intermediary filaments you will observe mostly in the nucleus. okay, and they are made up of lamin proteins. These are solid unbranched filaments formed of variety of proteins. If you can see, functions include formation of nuclear matrix, and this supports the support of membranes, supporting array of cytoplasm, and this supports contractile units, mainly mechanical strength to the nervous tissue. Okay. So, this is the complete comparative analysis of microtubules, microfilaments and intermediary filaments. And then here you see, now we will look into the double membrane bound organelles. So, amongst double membrane bound organelles, first let us look into the mitochondria. Mitochondria, this is the structure of your NCERT beta, but then let us draw a cartoon structure here to analyze the structure more keenly. Okay. So, this is the outer mitochondrial membrane, and the infoldings of inner mitochondrial membrane is what ma? Cristae. Cristae or inner mitochondrial membrane. The protein and lipid composition in the outer mitochondrial membrane and intermitochondrial membrane are different. In outer mitochondrial membrane, the protein composition is 60% of proteins and 40% of lipids. Okay. In inner mitochondrial membrane or cristae, it has 80% of proteins and 20% of lipids. Okay. And the space in between outer mitochondrial membrane and inner mitochondrial membrane is what? Intermembrane space. Okay, this space is called as intermembrane space, or you can also call as perimitochondrial space. Mitochondrial space. Fine. And inside the inner mitochondrial membrane, what is present beta? Mitochondrial matrix. Mitochondrial matrix. Fine. Now, what are composed in inner mitochondrial membrane and mitochondrial matrix? You have to know that. All the enzymes, electron carriers, whichever are required for electron transport system are present in the inner mitochondrial membrane. Okay. Take down all the enzymes and electron carriers required for ETS are present in the inner mitochondrial membrane. And mitochondrial matrix will possess what? ATP, that is the cellular energy of the cell, which is the energy currency of the cell, is present in the mitochondrial matrix, and it has a double stranded circular DNA. Mitochondrial matrix will possess what? Double stranded circular DNA. As mitochondria and chloroplast also possess double stranded circular DNA, they are called as what? Semi-autonomous cell organelles. Semi-autonomous or self-duplicating cell organelles. Semi-autonomous or self-duplicating cell organelles. Clear, right? Outer mitochondrial membrane, inner mitochondrial membrane, intermembrane space and mitochondrial matrix. You have to be very clear about the steps of aerobic respiration as well, because they will be asking you the questions on locations beta. In aerobic respiration, you know, glycolysis is the common pathway for both aerobic and anaerobic respiration. And in aerobic respiration specifically, the next two steps which are happening, that is oxidative decarboxylation and tricarboxylic acid cycle, that is Krebs cycle, both are operated in the mitochondrial matrix. And electron transport system is operated in the inner mitochondrial membrane. So, those 2 points are important about locations. Okay. In aerobic respiration, in aerobic respiration? First thing is what? Both oxidative decarboxylation and TCA cycle are being operated where beta? In mitochondrial matrix. Fine. And then electron transport system, last step of aerobic respiration, that is being operated in inner mitochondrial membrane. And inner mitochondrial membrane will also have some particles called as F0, F1 particles or ATP synthase particles. Okay. So, what happens is, let us suppose this as outer mitochondrial membrane and this as inner mitochondrial membrane. Inner mitochondrial membrane, I told you, right, what are present? F0, F1 particles or ATP synthase. So, here if you see, they have what, ma? Yeah, F0, F1 particles are also called as oxysomes. What are they called as? Oxysomes. So, here if you see, F0 is embedded inside the inner mitochondrial membrane, F1 is projected towards the mitochondrial matrix. Very important point you have to note. When we are dealing with ETS, you will know why it is projected like this, to promote the, it is the result of what? Proton gradient that is generated in between inner mitochondrial, intermembrane space and mitochondrial matrix, of which complex 5, which is called as ATP synthase, or F0 F1 complex, or it is also called as F0 F1 complex, ATP synthase, and Fernandez-Morgan particles also we will call it as, not required, you can take oxysomes. Fine, which will pump the protons from intermembrane space to mitochondrial matrix. Fine, protons are pumped from intermembrane space to mitochondrial matrix. Clear? Understood? Locations are very important, ma. Next is about chloroplast. Chloroplast is again double membrane bound organelle. So, when we have to deal with double membrane bound organelles, firstly you have to know this thing ki plastids. Plastids are the double membrane bound organelles. All the plant cells will have plastids. Plastids are of 3 types. What are they, ma? Plastids are of 3 types. What are they? First is chloroplast, chromoplast, good, chromoplast and leucoplast. Fine, right? So, chloroplast, these are green colored plastids, they have abundant pigment, that is chlorophyll. Fine. In some unicellular primitive green algae like Chlamydomonas, chloroplasts are 1 per cell beta, whereas in mesophyll cells you can see 20, 30 to 40 chloroplasts per cells. Okay. So, it can be 1 per cell in example Chlamydomonas, or 20 to 30 chloroplasts per cell you can see, for example, in a mesophyll cells. And their shape is also different. Chloroplast shape can be discoid, oval or cylindrical, it can be in the mesophyll cells, or you can see cup shaped chloroplasts in Chlamydomonas, and discoid shaped in Chara, spiral shaped in Spirogyra. Ribbon shaped in Edogonium. And next is chromoplast. Chromoplast is the one which gives different colors, right? Okay, so the color is based on the pigment. Fine. Colored pigments, it can be fat soluble pigments or water soluble pigments. Two types of pigments are there, ma. Fat soluble or water soluble pigments. Fat soluble or water soluble pigments. So here if you see, fat soluble pigments include what? Anthocyanins, xanthophylls, and carotenoids. Fine. Water soluble pigments include phycobilins and phycoerythrins. Any doubt, Abdul? Phycobilins and phycoerythrins. R phycoerythrin you will see in which algae? Red algae. Good. Examples of red algae? Porphyra, Polysiphonia, Giladium, Gracilaria. Good. Next is leucoplast. Okay, leucoplasts are colorless storage plastids. Okay, these are colorless and they store food material. If they are storing oil, oil is the storage body, then it is called as what, ma? Eleoplast. Starch is stored, it is called as what? Yes, amyloplast. Fine. And if lipids or oil is one and the same, ma? Carbohydrates or starch, one and the same. Protein. Good. Aleuroplast. Clear? Fine. Take down quickly and then we will look into the chloroplast structure in detail. Yeah, next. Here you see chloroplast structure. Chloroplast is a double membrane bound organelle again. Okay. Let us again take one cartoon diagram. You will understand easily, no? That's why. Okay. So here it has again outer mitochondrial outer membrane, outer chloroplast membrane and inner membrane. Okay, the space between outer membrane and inner membrane is called as what? Periplastidial space. Periplastidial space. And fluid filled space inside the chloroplast is called as stroma or chloroplast matrix or matrix of the chloroplast. Okay, this is called as what, beta? Stroma. Stroma or matrix of chloroplast. Stroma or matrix of chloroplast. So here if you see, there are membranous sac-like structures. Membranous sac-like structures inside the chloroplast. These membranous sac-like structures are called as what? Thylakoids. Okay, membranous sac-like structures are thylakoids, and the membrane of the thylakoid is called as thylakoid membrane or thylakoid lamella. Okay, the membrane of it is called as thylakoid membrane, and or thylakoid lamellae. Fine, right? And the stalk of sac-like structures, here can you see the stalk of sac-like structures? The stalk of sac-like structures are called as what? Grana. Fine. one grana is connected to other grana by what? Stroma lamellae. Can you see these connections? Yeah, this is called as stroma lamellae. And inside the, yes, that I will tell you, have patience. Okay. So, this matrix or stroma, which is the fluid filled space, it has what? Again double stranded circular DNA, it has starch granules and lipid droplets. Okay. Stroma is having what, ma? Yeah, stroma is having a double stranded circular DNA. Yeah, double stranded circular DNA, starch granules, yeah, starch granules and lipid droplets. Now the question is about this double stranded circular DNA. Double stranded circular DNA, whichever is present in the matrix of the chloroplast, this is involved in the synthesis of world's abundant protein, that is Rubisco. Fine. Always remember beta, the ultimate goal of DNA is to synthesize a protein. Okay. And here in the chloroplast, it is synthesizing the world's abundant protein, that is Rubisco. Fine. This Rubisco is catalyzing the activity of what? Dark reaction again. So, this is the structural description of the chloroplast that you have to know. And as both chloroplast and mitochondria are possessing double stranded circular DNA, double stranded circular DNA is a characteristic feature of what? Prokaryotic cell. And it is also having 70S ribosome, that is also a characteristic feature of prokaryotic cell. It is possessing the features of a prokaryotic cell because both chloroplast and mitochondria are once upon a time they are prokaryotic cells actually. In due course of evolution, due to the process of endocytosis, they have been entered into the eukaryotic cell and they have become part and parcel of the eukaryotic cell. Okay? That is clearly explained under the concept of endosymbiotic theory. So, with the evidence of endosymbiotic theory, we can say that mitochondria and chloroplast are once upon a time prokaryotic ancestors. Okay? Take down that also. Yeah, I will write, wait. Okay, it is endosymbiotic theory beta. Endosymbiotic theory, which gives you the proper evidence on what chloroplast and mitochondria were prokaryotic ancestors. And due to the process of endocytosis, they have become a part and parcel of eukaryotic cells. Fine. And now they are independent in living, yes or no? Chloroplast and mitochondria. Mitochondria is called as powerhouse of the cell because it synthesizes energy. Even chloroplast is called as kitchen of the cell. It synthesizes food and energy as well. Okay. Next. Now you are clear with the chloroplast. About nucleus. See, nucleus is having 4 components inside it. What are the 4 components inside the nucleus? First is nucleolus. Raghav, you asked me that question, right? We will finish one more thing about the events of photosynthesis. Fine. Photosynthesis is conducted in 2 sequential events. One is called as light reaction. Just a second. Take down parallelly. Light reaction or light dependent reaction also we will call it as. And second is dark reactions, which are rightly called as light independent reactions. Fine. Calling light independent reactions as dark reaction is a misnomer, but then it is noted like that. So, what are the 3 events of light reactions beta? First, it is absorption of light in the form of photons. Absorption of light in the form of photons is observed in the site of this reaction is thylakoid lamellae or thylakoid membrane. Okay? Because in thylakoid lamellae photosystems are present. Okay, you know, right, PS1 and PS2 we have discussed in photosynthesis. Fine. These photosystems are present in thylakoid lamellae. So, absorption of light in the form of photons happens in thylakoid lamellae. Second is what? Yeah, good. Photolysis of water. Photolysis of water. This photolysis of water happens where ma? Inside the thylakoid lumen. Fine. Or you can also say in the inner membrane of the thylakoid. Inner membrane of thylakoid. This is also correct because adjoining to the inner membrane of the thylakoid, oxygen evolving complex is present, which is facing towards the thylakoid lumen, that is being doing this activity of photolysis of water. And third event is what, beta? Third event is formation of assimilatory power. Assimilatory power, that is ATP and NADPH. Fine. This assimilatory power is utilized to fix what? Carbon dioxide to glucose in chloroplast stroma. Formation of assimilatory power takes place in chloroplast stroma only. Okay? And this ATP and NADPH are utilized to fix carbon dioxide or for the formation of glucose. This happens where again in the chloroplast stroma. Clear? Clear, Raghav, with the locations? Inner mitochondrial membrane, oxygen evolving complex is present, which is facing towards the lumen. Exactly here this takes place. Okay? And next, see here, nucleus. It has 4 components, right? What are they, ma? Nucleolus. Second is nucleoplasm. And third is nuclear envelope. Fourth will be nuclear pores. So, nucleus is surrounded by a nuclear membrane which is double membrane bound. Fine. Inside the nucleoplasm, you have a nucleolus. Nucleolus is purely involved in the synthesis of rRNA, beta. Nucleolus is purely involved in the synthesis of what? rRNA with what polymerases? RNA polymerase 1. Fine. And here in the nucleoplasm, both the synthesis of, inside nucleoplasm, synthesis of mRNA and synthesis of tRNA will happen. Okay? Synthesis of mRNA and synthesis of tRNA. mRNA by RNA polymerase 2, tRNA by RNA polymerase 3. Fine. This is by RNA polymerase 3. So, here inside the nucleoplasm, along with mRNA and rRNA synthesis, what is happening? Synthesis of, what is happening? A presence of genetic material is also there. That is, linear shaped chromosomes are present. Fine. And then nuclear envelope extensions will lead to what? Endoplasmic reticulum. Those which has ribosomes, it is RER. Those which does not have, it is smooth endoplasmic reticulum. And then it also has what, beta? Nuclear pores in between for exchange of materials in between the nucleus and cytoplasm. The materials can be RNA particles, proteins, mRNA. Fine. These are being exchanged from nucleus and cytoplasm. This is the structural description about the nucleus. Now, we need to highlight the structure of chromosome. Okay, structure of chromosome. Look at here. So, this packaging is not required right now. Look at this chromosome structure. This diagram which is given in your NCERT is showing first primary constriction. This primary constriction is called as what, ma? Centromere. Centromere or primary constriction. this primary constriction adjoining to this two chromatids are present. Okay, this is one arm of the chromatid, this is the other arm of chromatid. So this is one chromatid, this is the second chromatid. How many chromatids are present, beta? Two chromatids. Two chromatids. Okay, in few chromosomes, in most of the chromosomes, there is a secondary constriction which is also present above the chromosome, above the chromatid arms. That secondary constriction is called as satellite DNA. Satellite DNA, which has repetitive DNA sequences, which is used in DNA fingerprinting or polymorphism. Fine, understood? So this is what primary constriction. To this primary constriction, two disc shaped structures are present. This disc shaped structures are called as what, ma? Kinetochores. To this kinetochores only spindle fibers are attached. Spindle fibers are attached during the time of cell division. Okay, spindle fibers attached to which part of the chromosome? Kinetochores. Kinetochores are disc shaped structures present attached to what? Primary constriction, that is centromere. Secondary constriction is satellite DNA. So here in this chromosome structure, how many primary constrictions are present? Primary constrictions are centromeres.
Only one.
One. Fine. How many chromatids?
Two chromatids.
Chromatids are two. How many arms of the chromatids are present, beta? Four arms of the chromatids. How many secondary constrictions?
Four.
Hai, see and say re.
Two.
How many secondary constrictions?
Two.
Two secondary constrictions. How many kinetochores?
Two. Only one disc.
Yeah, kinetochores are how many? Two. Okay. Now let me draw one more structure of the chromosome, ask you for the number of chromatids. And you are clear with ploidy, no? What is haploid and what is diploid condition? What is haploid and diploid? If you don't know haploid, diploid, it is difficult to understand sexual reproduction of flowering plants, cell division and genetics as well. Haploid and diploid condition means what? What is ploidy? How you can decide the ploidy?
Two chromosomes are bonded on haploid and
Diploid to an haploid, and that's all we know, right? See, human cell is having how many chromosomes, Nana?
Two.
Like
46.
46 chromosomes. How many pairs?
23.
23 pairs of the chromosomes are present in a human cell. Okay, so this is the complete set of the chromosomes. What is the complete set? 46 chromosomes or 23 pairs is the complete set of the chromosomes. Half of this is how much? 23 chromosomes is half of this chromosomes. Okay, half of the 46 are 23 chromosomes. So if at all in any cell half the set of chromosomes are present, then it is called as haploid. Complete set of chromosomes are present, so it is diploid. Okay, you don't just think this is haploid and this is diploid. No. Okay, now I will give you a clarity on polyploid condition. What is polyploid, beta? Polyploid means a single centromere having many chromatids like this is polyploid condition. Polyploid condition is observed. Polyploid condition is observed due to sequential karyokinesis but no cytokinesis. Nucleus is being dividing, chromosomal division is happening, but then there is no cell division. It will lead to what? Polyploid condition. Understood? Got the difference? Sequential karyokinesis, complete karyokinesis division, but no cytokinesis is happening, then polyploid condition will appear. Okay, got the difference between haploid, diploid, polyploid, okay, and about chromosome structure also. Now you see shapes of chromosomes. Shapes of chromosomes. So here the shapes of chromosomes, here you see, one thing you have to know during the time of cell division also, during metaphase of cell division you can clearly see the structure of the chromosome. Okay, take down, important point actually, during metaphase of cell division, you can clearly see the structure of chromosome. Yeah, okay. And here if you see anaphase, during anaphase of the cell division, yeah, shapes of the chromosomes are clearly observed. So under microscope, if you want to clearly see the structure of chromosome, under which phase you have to see? Metaphase. If you want to see different shapes of the chromosomes, under which phase? Anaphase. Now clearly you observe this, ma. So when centromere splitting is happened and two chromosomes are moving to the opposite poles, they can move in different shapes. If two arms of the chromatids are equal and centromere is at the center, that moves in the form of V shape like this. Okay, two equal arms with V shape. Okay, this is called as what chromosome? Metacentric chromosome. Metacentric chromosome. And centromere is towards, slightly towards one end, which is resulting in one short arm, one long arm. This is called as what chromosome, beta? Submetacentric chromosome. Submetacentric. Submetacentric is what shaped? L shape. One long arm and one short arm. Fine. And lastly it is acrocentric chromosome. Acrocentric chromosome, where centromere is very close to one end, which is resulting in one extremely short arm, one extremely short arm and one long arm. This shows what shape? J shape, extremely short and one long arm, very long arm. Okay. Next, last is telocentric chromosome, where one arm is very negligible and other you can see very long arm and centromere. Fine. So this will result in what shape? I shape. This is telocentric chromosome. Fine. So clear with shapes of the chromosome? Are we missing out anything? Yes, we are. Microbodies we have to deal, no? Single membrane bound organelles we have dealt with ER, Golgi, lysosomes and vacuoles. Microbodies are left over, right? So here you see, microbodies are single membrane bound organelles, small organelles which are present in the cytoplasm of both plant cells and animal cells. So they are present both in plant cells and animal cells. They absorb molecular oxygen for oxidation. The main purpose of microbodies in the cell is oxidation of long chain fatty acids. Okay. So microbodies are of two types. What are they? Peroxisomes and glyoxysomes. So here if you see, peroxisomes and glyoxysomes, both are single membrane bound, right? Both are single membrane bound. Peroxisomes are present both in plant cells and animal cells, whereas glyoxysomes are present only in the germinating oil seeds. Why? What happens in glyoxysomes is glyoxylate cycle will operate, beta. Glyoxylate cycle. Okay, where oil seeds will be having fats or lipids. Those are being converted to simple glucose molecules by this glyoxylate cycle. Okay, and they are also present in fungal cells and oil seeds. And then what are the key functions of peroxisomes and glyoxysomes? Most important. Okay, the key functions include from peroxisomes though fatty acid oxidation, lipid synthesis, detoxification of drugs, glyoxylate cycle and photorespiration. You have very keen idea about photorespiration, right? Next, and here glyoxysomes will contain the enzymes for beta oxidation of fatty acids, and here also glyoxylate cycle will be operated in oil seeds. Okay, and then we have role. What is the role of peroxisomes? General metabolic and detox roles are present in all cell types, and here in glyoxysomes lipid metabolism is taking place during seed germination. Fine. So about complete chapter, is it clear to you? Once you recall patiently what we have dealt from the beginning: cell theory, cell discoveries, shapes of the cells, different sizes of the cells, then prokaryotic cell, okay, its structure and physiology also we have discussed in reference with cell, okay, and its structure, shape we have discussed everything, motile and non-motile structures, fine, sexual recombination, conjugation, transformation, transduction we have discussed, useful and harmful moneyrons we have discussed, and then we have looked into eukaryotic cell, clear differences between plant cell and animal cell, then cell wall, cell membrane, fine, and then about endomembrane system, fine. What are the organelles involved in endomembrane system? Endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. Post that we have discussed about non-membrane bound organelles like ribosomes, which are present both in prokaryotic cells and eukaryotic cells, centrioles which are present only in the animal cells, and then we have dealt with cytoskeleton, learnt about comparative analysis of microtubules, microfilaments and intermediary filaments, and then we have looked into double membrane bound organelles, chloroplast, mitochondria and nucleus. Lastly about microbodies. With this the chapter is done beta. In our next class we will be starting with cell cycle and cell division. Okay, thank you.